Hello, thank you for the opportunity to speak today. Today I'll be introducing some research at the interface of two important fields, biodiversity and human health. This work represents somewhat of a starting point for a larger body of research, ultimately aiming to build the evidence base for example, nature-based health policy and, for example, supporting the design of new biodiverse green spaces, with overall objectives to help benefit both environmental management and human health. This work is supported by JUNA and sits jointly in the Schools of Biological Sciences and Public Health at Adelaide Uni, under the supervision of Phil Weinstein up the back there, Peng B and Michelle Waycott in JUNA, and Peng's with Public Health. Get my clicker out. So this graphic here is taken from a 2015 World Health Organization report appropriately titled Connecting Global Priorities, Biodiversity and Human Health. The branches of this tree represent various mechanisms and co-benefits linking biodiversity and human health. You'll recognise some familiar themes in here. Food and water security, climate change, infectious disease control and the like. There's one link here, microbial biodiversity, is among the least understood and therefore represents a research gap, while also possibly having the largest background influence on human health, simply because microbes are everywhere. This mechanism is probably best explained by the concept of microbial old friends. So and we've got some old friends here just for a bit of fun. Over evolutionary timeframes, humans have been exposed to all manner of muck and grime and microbial diversity in our surroundings. We've developed a relationship with certain microbes where they play a key role in stimulating, educating and regulating our immune system. This involves a balance between triggering inflammatory responses to shut down pathogens and, on the other hand, suppressing unnecessary and damaging responses to normally harmless allergens and the host's own tissues and mild pathogens in food or from the surrounding environment. And it's this role of microbes in teaching our immune systems when not to go on the attack that medical researchers are increasingly interested in. So there's this notion of certain microbes being old friends with humans because of this evolutionary relationship that's formed between us. They may be mild pathogens or commensal organisms, which are otherwise thought to not help or harm us. They overlap with species found in the normal human microbiota. Microbiota means microbial communities, and these are found, on the skin, in the airway, in the gut, and these microbial old friends can originate from the natural environment. So these ideas first gained attention under the banner of the hygiene hypothesis, with the notion that we could boost health through some regular dose of the right microbes. So environmental microbiota supplement and interact with the human microbiota. In this graphic, their influence might be represented by this green shading here. So they interact with our normal microbial communities on the body, and with all other factors being equal, such as diet and antibiotic use, you end up with a well-balanced microbial system in your body. Whereas without that sort of appropriate contact with environmental microbes feeding into that system, dysbiosis can result. So this means an imbalance or a reduced diversity in the human microbial communities. And this is, apart from immune system dysfunction, this is also linked to a range of diseases such as diabetes, obesity, depression and even some cancers. So existing research tells us that different types of environments, different soils, different plants have their own characteristic microbiota associated with them. So basically a loss of landscape scale. So there's the microbial habitats are all different with different types of plants. So when you lose basically that landscape or macro scale diversity, this translates into a loss of microbial diversity. Animals also have their own characteristic microbiota. So the science of aerobiology, which is biology in the air, tells us that the air is alive with all manner of microbes, bacteria, viruses, fungi, algae and the like. So now we're talking about a real biological connection between our environments and the human health. Non-living components. Non-living components, so not just microbes, we're also talking about things like volatile organic compounds, endotoxin, which is a breakdown of decay products of microbes. Air ions are also in the mix of influencing immune system. And this is not totally new. I guess for over a couple of decades, Japanese scientists have been studying this thing called forest air bathing. It sounds a bit way out, but they have been looking at these unseen agents acting on human health that are not just simply the absence of toxic urban air. But key questions remain. What types of environments are most beneficial? What exactly are these microbial old friends? And is it microbial diversity that is required? And what dose or exposure is required to see the benefits? So big picture, you need a multidisciplinary team is needed to draw all those connections between the environments, their microbiota, people's exposure to this, whether there's direct or indirect influences on the immune system or modulating the immune system, and through to health outcomes. This seems like an overwhelming challenge, and where would we start looking? So my piece of this puzzle is basically, just part of this overall puzzle, looking at a high level to, focus where we're going to look, where are the best, most cost-effective places to start looking into this stuff. So my background is in digital soil mapping, and that involves environmental correlation analysis, so that's basically what I'm sort of bringing to this. Looking at environmental correlations between different types of environments and those sort of health outcomes on a spatially represented relationships. Yeah, so why treating these as environmental proxies for those underlying microbial mechanisms? There's just so many unknowns at the moment that where would you know where to start? A bunch of different expertises on their own feeding into this, drawing this big connection. So I guess my point of view is if we can wrap up these unknowns in something a bit more familiar as a potential starting place to look at these influences, then this will be of value on its own, but also provide a focus for those more detailed efforts. So the hypothesis that's the study of landscape scale environmental proxies can inform and prioritise further research into these protective environmental microbiota mechanisms. Because this, I believe, is where the research gap is in this whole biodiversity and human health space at the moment. Okay, I mentioned the question of dose before. So this is a hypothetical graph of health impacts, plus and minus on the y-axis, increasing dose of a theoretical microbial old friend on the x-axis here. So this relationship has to join the traditional microbiology view where, so say E. coli is in this mix of a microbial old friend. So more and more E. coli, you've got to have a downward trend in health down there. But Microbial Old Friends says, hey, we need a bit of this stuff for triggering our immune system. And in the middle there's some sort of happy medium or Goldilocks zone where things are just right. So maybe this is obviously at a human scale, but perhaps these are reflected in, say, some sort of soil microbiology index where there's different levels of exposure to this type of thing. Probably just skip over that. So what I'm going to be doing is taking Public Health Information Development Unit Health Atlas data, which is social and health stats by local government areas. They have other spatially distributed values. That'll be my target sort of areas of health with some key social indicators in there as well. Running, basically gathering as much sort of environmental data as I can, because the environment is such a multifaceted thing that the more information you can bring into this, natural experiments, you can't just sort of pick on just a few drivers. So I'll have a bunch of numerical layers. A lot of these are widely available now or publicly available now. For categorical, some key categorical layers that I want to bring in, and I've been doing a focal raster calculation on a 3 kilometre radius, say for land cover classes and vegetation mapping classes and things like that. There's not true sort of measures of biodiversity, but a diversity index of Shannon's diversity index over a focal area, and also class proportions for different land cover classes, land use types, vegetation class mapping and things like that. So some statistical analysis. I guess I wanted to bring in something that's a bit more interpretable than your traditional principal components analysis, where, like, there was a study in Finland where they looked into some of this stuff and they said they couldn't really separate three principal components. They couldn't separate the influence of forest and agricultural land. And from a environmental viewpoint, we'd like to know that they perhaps have a different influence. So this thing called a LASSO, or least absolute shrinkage and selection operator, you can Google that if you like. It's like a machine learning version of a multiple linear regression. So the results are a lot more interpretable than some of your other methods. I also have to bring in some more standard measures of significance to make it sound convincing. So across Australia, local government areas. So some preliminary results. We've got 4 different health outcomes here. I think all with and the idea of this is having a plausible linkage mechanism, a plausible microbiota linkage mechanism. So digestive disease, infectious and parasitic disease, cerebrovascular disease, which includes stroke, and stroke has a component of inflammation in there. All cancers, public hospital admissions. So 3 public hospital admissions, 1 death here. So on the bottom here we've got increasing land cover diversity, where the Shannon's index. So all downward trends in terms of improved health outcomes with this land cover diversity index. It looks interesting. It's, as I said, big picture. And when socioeconomic status is sort of brought in, these still remain some level of significance in a sort of a 2-variable thing. So just another quick one, some of the LASSO results. So this is a respiratory health disease across those local government areas. So of 100 social and environmental variables fed in, this thing I ran through a 10-fold resampling and modelling to generate a bit of uncertainty in the analysis. So we've got 25 indicators, so 25 potential drivers coming in here with the usual suspects that you'd want to see. So socioeconomic is right at the top. We've got percent Aboriginal persons in there, and we know that they have health problems, unfortunately. And there are some climatic drivers in there that are known to be associated with respiratory health. But I guess in this mix, and this tool has allowed us to sort of see side by side, perhaps there's something associated with these environments that is interesting and could be contributing here. So we've got distance to coast, and there's microbial influences. Being close to coastal, perhaps it's a dryness or aridity. So we've got to look at, it's something to look into further. Species richness from the Atlas of Living Australia is right up there. So, and then we've got sort of some of the fractional cover things where these are kind of ranked by, I didn't explain, by the size and sign of the standardised regression coefficients. So you've got basically the bigger it is out on this way is a bigger effect. And if it's on this side, it's kind of viewed as a protective, and on that side, it's an adverse thing. And on that side, the bars are basically how many times it's cropped up within each of the 10-fold runs. So bigger effects up the top, more often, all the time or more often in terms of the bars there. So anyway, some things to sort of look into, and it is supporting the hypothesis. So there's a bunch of limitations to this approach, which I don't really have time to go into. So it's about identifying and prioritising those environmental biohealth relationships that are worthy of more detailed investigation. I think it's going to build insight, and we'll get some new methods and tools from this. And it's all about supporting that evidence-based policy to get those win-win outcomes for biodiversity and health. So.
Thank you, Craig. We have time for one very quick question while the next speaker gets on board.
Yeah, in your modelling and comparison of the land cover versus the health, did you look at concrete as a cover or just use vegetative covers?
So, yeah, some of the land covers were Geoscience Australia land cover classes, so it's 250 metre resolution. They do have urban in there as a land cover class. Yeah, and so that kind of, it didn't pop up out in the results, but yeah, urban areas. I had another layer for distance to built up areas. That didn't really show up, so yeah. This thing's quite selective, it kind of pulls things out, so.